Hello, good evening, everybody. May I ask you to take your seat? I have just been informed that we're already live streaming, so it's time to get started. Welcome, everybody. Welcome to Algonquin College. Welcome to the Brain Health Awareness Week, hosted by the University of Ottawa Brain and Mind Research Institute. My name is Michael Schlossmacher. I'm the interim director of the Brain and Mind Health Research Institute, and it's a real pleasure to welcome all of you. We have a wonderful program tonight, and as you know, the title of this evening's event is Coming Together to Care for the Aging Brain. And the organizers of this evening's events, in part from the Elizabeth Bruyère Hospital and the University of Ottawa, and the two partners formed a collaborative effort a year and a half ago that calls itself the Memory Collaborative Group. And it's that group that has put on the program for tonight. And as you know, the topic is of great interest to not just the people here in the room, but society in the Western Hemisphere in general. As our populations age, the rate of cognitive difficulties, memory problems, and dementia are increasing. And so this is a really important topic that's not only dear to the specialists, but also general practitioners. And there's not a family in the Western Hemisphere that's not affected in some fashion by thinking difficulty and memory difficulty. So we're going to start off the evening with a brief video that summarizes what the Memory Collaborative Group is all about, as engineered by the Elizabeth Bruyère Hospital and the University of Ottawa. And then afterwards, I will introduce a special guest to you. And then thereafter, we'll have three outstanding speakers. Okay, so the video is next.
Collaborative is a unique partnership that brings together researchers from the University of Ottawa and researchers from the Research Institute at Bruyère to bring their expertise together to try to understand the fundamentals of memory and cognition. Memory allows us to be who we are. We remember familiar faces, find our way home, can speak a language. Memory is truly fundamental to our existence.
This is a really exciting time for us because now we're able to combine researchers doing lab research where they might study the movement of a mouse through a maze and compare that to clinical research, which is how does an older adult move through their apartment, to real life clinical experience, which is what I do every day at the clinic, which is decisions about safety at home.
It started about, I would say, 8 or 10 years ago that I began to notice, while I was still working, of course, that there were things that just didn't feel right. It's really marked a lot. I can't do much in terms of this is exactly a real good example of how I lowered myself up. But I have my wonderful husband here.
All of the efforts that we're putting in can provide a more streamlined approach to identifying, preventing, and treating disorders of cognitive decline. Right now, I really feel that we're at this critical point where we have the opportunity, we have the resources, we have the knowledge to get us there. And it's important that we involve a lot of people and we have a very interdisciplinary approach where we can take these pockets of research and communicate better so that we can find those cures, we can find those interventions.
But now, brain health has sort of been neglected maybe a little bit over the time, and I think people don't want to look at memory loss and brain health as an issue. There's now more people suffering from cognitive decline than there are having heart attacks, right? And I think what we need to do as a city is to get behind that and build up the critical mass of researchers and both primary researchers and clinical researchers that we can move this whole thing forward and develop between the University of Ottawa and Bruyère world-class brain health center here in Ottawa.
Definitely.
Where people can come to from all Eastern Ontario and feel as comfortable and as well looked after as we have.
Yeah.
Well, that was a wonderful introduction. Thank you for the team that put it together. And just want to introduce to you the concept as to how the Brain Health Awareness Week started 6-7 years ago. In the head of a gentleman and his wife, they're here in the room, Ucho and Sam Oosterhoff, who are community members and philanthropists. And they said to us 6-7 years ago, you're not doing a good enough job at the University of Ottawa. You're not talking about brain health enough to the community. You have to raise awareness and engage the community. And right they were. So 5 years ago, this enterprise began. Thank you very much, Ucho and Sam. And I remember Sam saying at that time too, and you have to get somebody who's a politician and declare the opening of this week. And you have to make it a big deal in the city so that people, our citizens, pay attention to it. So Sam was right. So we've had wonderful attendance by prominent leaders of our community. And today it's my special privilege to introduce Natalie de Rosier, who is going to help us open this evening. And she's a heavy hitter. She has many, many hats that she has carried over the years. And she is an attorney. She got her law degree at the University of Montreal. She pursued a master's degree in Cambridge, Massachusetts at Harvard University. She rose through the ranks. She was admitted to the bar in both Upper Canada and Lower Canada, worked as an attorney, and then dedicated her career to academic work, and she rose all the way to the ranks of Dean of the Law School at the University of Ottawa, so she's a colleague of ours, and made it even to run as an interim vice president for a year plus before dedicating her time and efforts to politics. She served in the previous government as a minister, and she's now currently parliamentary expert on issues regarding to housing and women's affairs, and her portfolio is actually much, much bigger than that. But it's a real pleasure that she takes time out of her busy schedule to come and join us tonight. Madame. Merci beaucoup. Bienvenue.
Thank you very much. And first of all, thank you, Utra and Sam, for being here, for having had the leadership to raise for Ottawa the idea of caring for the aging brain and talking about the healthy brain. I am very happy to be here with you today. It's a real pleasure. I want to acknowledge that Algonquin College is on Algonquin territory, and I think it's always important to remember where we come from. Not only did I have a great career in law before going into politics, and I am so glad to celebrate here the partnership between Bruyère, which is in the Ottawa Vanier riding, which is the riding that I have the privilege of representing, and University of Ottawa, which is so dear to my heart. As a dean, I had the occasion to begin a Center for Law and Health, where one of the issue was mental health. And I continue to think that this is the challenge of our generation: climate change and mental health. And on a personal note, my dad suffered of Alzheimer, and for the 7 years that he was diagnosed. So I know the impact that it has on the family. Of his family, there were 11 children. 6 of them died of a heart attack, and 5 had Alzheimer's. So I'm quite interested in the genetics aspect of the disease as well. But I know, because I have seen the impact that it had on my mother and on all of our family. And I've had, since then, the occasion to visit many of the long-term care centers that we have around Ottawa, and see the difficulty in caring caring with dignity of people who suffer from cognitive dysfunction, dementia, and Alzheimer, and also the impact that it has on the other patients and on the people who work in the institutions. So I think it is tremendously important that we continue to work on this, and we know that it's a problem that will keep growing with our aging population. So my task is not only to congratulate you on being here and on wanting to do this work and continue to do this work, and to celebrate the Memory Collaborative, the partnership between the University of Ottawa Brain and Mind Research Institute and Bruyère Research Institute. And by the way, I had the occasion to visit the little apartment that they did at the Bruyère Institute, where they actually use lighting to try to help patients. So it's quite amazing to see maybe technology could help us in some way solving the very difficult issues of safety. Mon rôle est également to read on behalf of Jim Watson the declaration of this week being, and it's going to be a real pleasure for me to read officially the Brain Health Awareness Week declaration. Whereas brain and mind-related illnesses affect 1 in 3 Canadians of any age, and whereas the socioeconomic cost of brain and mind-related illnesses is enormous in Canada and worldwide. And whereas Ottawa, led by the University of Ottawa and its affiliated research institutions and hospitals, is a national hub for brain and mind research. And whereas the University of Ottawa and its partners intend to improve the brain health of its citizens and nationally promote brain health. And I'll read a little bit in French. Attendu que la semaine de sensibilisation à la santé cérébrale visera à sensibiliser davantage la population aux maladies liées au cerveau et à l'esprit et à réduire les préjugés qui leur sont associés, whereas Brain Health Awareness Week will aim to improve public awareness and reduce the stigma associated with brain and mind-related illnesses. Therefore, and it's written by Jim Watson, but I'm particularly happy to say that I hereby proclaim October 15 to October 19th, 2018, Brain Health Awareness Week in Ottawa, and call upon all citizens to mind their brain. Merci beaucoup.
Merci.
What a beautiful, libérer le pouvoir du cerveau. What a great...
Merci beaucoup, Madame. Vous étiez très gentille. C'était un grand honneur pour nous. May I ask the speakers to come up here and we'll take a photo.
Yeah, it's your
Maybe we'll have you in the center, Madame.
Yes. Okay, come in. Well, congratulations.
Thank you.
Bravo.
Thank you, Madame. Thank you. Thank you so much. Okay. Wonderful, thank you. Let's get started with our first speaker, Dr. Natalia Cheburashka. It's a wonderful opportunity to listen to her. She's an assistant professor. Dr. Cheburashka is an expert. She works at the Institute of Mental Health at the Royal Ottawa Hospital. She's associated with the University of Ottawa in the Faculty of Social Sciences and also with the Department of Cellular and Molecular Medicine at the Faculty of Medicine. Her interest is at the interface of depression and memory function, depression in young people as well as in adults. And she's using different markers, different approaches to ascertain how well people respond to antidepressive treatment. And she's using sophisticated machinery, brainwave recording, complex brainwave recording and its interpretation together with brain imaging. And she's going to talk about memory. What exactly is it? And so let us welcome Dr. Natalia Cheburashka.
All right. Can everybody hear me okay? Wonderful. So I do feel like a little bit of a hack because, as was mentioned, I mainly study depression. But nevertheless, as I was explaining to a handful of individuals, depression and actually a lot of mental illness is also accompanied by a lot of cognitive symptoms. So what we get is people who recover quite well from their depressed episodes, for instance, yet they come into our lab and say, you know what? I still can't concentrate. My memory is still not great. So the interface between mental health and cognition is so intimate that I'm feeling a little bit better coming up here and talking about memory to you guys because I think I know something. So basically, I would like to give you a synopsis of the various types of memories that exist, some of the neurological or biological underpinnings as to what is a memory, and then just briefly talk about forgetting, and some of the things that we can do that are sort of science-informed that we know does help us with our memory. So this is a little bit of a complicated picture, but what I'm trying to show here is that memory is not a unitary phenomenon. So there are different types of memories. We know that there's something called sensory memory. We know that there's short-term memory as well as long-term memories. I'll explain the short and long-term memories in a little bit more detail next, but I just want to draw your attention to sensory memory, which is effectively a buffer system of the stimuli that our brain takes in. So it could be auditory information, it could be visual information. We take it in very, very quickly, so it's not a memory trace that exists within our brains for long, yet it informs the subsequent development of short-term memories and long-term memories. So we can't really talk about memory without mentioning learning, because the two go hand in hand. You first need to learn something before it becomes a memory. So what we do is we acquire knowledge, we learn, generally through our senses, though of course we can also have internal learning if we ruminate on something, for instance, and then that gets transformed to our short-term memory. However, if we don't do anything with that memory, it does get lost. So we need to transform it into a longer-term memory. And that process, this kind of crystallization, this solidification from short-term memory to long-term memory, is something called the consolidation of a memory, or just simply consolidation. Now, as psychologists, we use a lot of theoretical frameworks, all scientists do this, in order to help us sort of distill complex ideas into something that we can grasp. And generally, when we are talking about short-term memory, or a form of short-term memory called working memory, we have kind of a theoretical framework to make sense of it. Now, what is working memory? I know I'm throwing so many definitions at you guys today. Well, working memory is sort of that active memory that stays in your head that you need to manipulate. So it's drawing various sources of information from various memory stores, from our sensory processes, and we have to work with it. A really good example is, okay, what's that phone number, 455? Okay, that's working memory. You're kind of manipulating it actively. And what we have in this memory system is essentially a temporary storage of visual information or verbal information, and that gets transported to something called the central executive. So this is our framework, and that central executive sort of organizes that information into something a little bit more coherent, a little bit more knowledgeable that we can work with. And then we can then move that memory in and out of our long-term storage. So that's sort of the framework for working memory. Well, that's nice. That's nice to have frameworks, but does it actually relate to the brain? Well, it actually does. We know that regions that are more anterior, so frontal of our brains, they're very involved in cognition. We use that frontal region to manipulate information, to inhibit our responses. It's a really critical component in cognition. But we also know that more visual types of those immediate memories, those sensory memories, they're processed a little bit further back. So even though we have these theoretical frameworks, they do have sort of a neurological basis to them. Now, declarative memory is a form of long-term memory. So it's a branch of explicit memory. We can skip that. Declarative memory is basically a memory that we can state. It's a conscious memory. And there's two types of these memories. Something called episodic memory, which, as the name suggests, they're episodes about our lives. We don't really need to put in a whole lot of effort to remember what happens to us. It's something personal. So you think back, oh, that was my, 21st birthday party. I remember what occurred there. While semantic memory, well, that's the memory that's a bit more factual. That's the one that you really need to work a little bit on to actually get it in your brain. So it's essentially our memory for facts, such as, well, we know that there's a municipal election next week and we should probably go out and vote. That would be an example of a semantic memory. Now, another form of long-term memory is implicit memory. So this is a form of memory that is less conscious, and it's generally tested. And a really nice example of an implicit memory is tying your shoes. Now, try explaining to somebody how you tie your shoes. Well, you make one. It's ridiculous, right? You simply know how to do it. So these are these kind of skills that we acquire and we get very good at, but we're not really conscious of exactly how those pieces come together. Another form of implicit memory is priming. So you can see that, for a lot of us, that middle camel, we can kind of make out that it's a camel, maybe with a little bit of practice. So that's kind of these cues of an incomplete stimulus that suggest, aha, we know what this is. Now moving on to my more favorite part of this talk, which is the brain, and really what are the neural mechanisms underlying memory. So I'm not going to do it justice in a couple of minutes, but long story short, our brains, as I'm sure you all know, are made up of cells which are called neurons, and those neurons are connected. And really what occurs when we are forming a memory, at least this is one of the theories of memory formation, is that effectively, neurons are all interconnected. And when we present a stimulus, such as the circle in the bottom, a certain proportion of those neurons that are interconnected will get activated. Now, if I keep on presenting a circle over and over and over again, that activation really gets strengthened. So there's an internal representation in our neurons of that circle. And in fact, what is very cool is that at one point, you don't even need the full circle. You can just have half of that circle, and because those connections have been strengthened, they're able to sort of create that representation of a circle for us. So there's a beautiful quote by Dr. Donald Hebb, who's a very famous Canadian psychologist, which is still used today because it's true. Neurons that fire together, wired together. And it's a really kind of helpful way of understanding a really complicated process. If we want to get down into a little bit more detail, the connections between these neurons are called synapses. And if we want to distill this into somewhat more molecular terms or biological terms, it's really the strength of the synapses that are summed together across a network that really in a biological way define a memory. I mean, it's sort of funny to distill something so beautiful like a memory, so complicated to this statement, but biology, it's a little cut and dry sometimes. But what about forgetting? We all know the importance of remembering, of memory. But is there any utility to forgetting? Well, actually there is. Forgetting too serves a function. And I think a really nice example of that would be something like post-traumatic stress disorder. That's really when a memory forms and it becomes so incredibly intrusive that it becomes debilitating and takes over essentially one's lives and has such a huge impact. So that's an example of something where forgetting isn't occurring as properly as it should be, and we know that there are certain neural features that allow us to forget. So the cells also have a way for those connections to become less strong, and that's really the basis of forgetting. Now, we do know with aging, and you guys will get a bit more details on this, our memory, unfortunately, does decline to a certain extent. In some people more, in some people less. Episodic memory, so actually those personal memories, on average show the most decline, which is a little bit surprising. Now, one of the theories behind that may be because there's a structure in our brains called the hippocampus, which is extremely important in memory formation. Unfortunately, it's also the structure that tends to be most likely to sort of deteriorate with age. It's also the structure that's most impacted by stress. So a really important structure, but one that's also very, very sensitive. So that might be one of the theories underlying this. Now, semantic memory, so that memory for facts is actually a little bit less impacted. Now, why is that? Maybe because we actually need to put an effort to remember facts. Maybe because we need to recall them more frequently on a day-to-day basis. That's just one hypothesis. Yet implicit memory, so that memory that you can't really articulate, that kind of subconscious memory, that's actually fairly stable. So adults, certainly older adults, continue to learn, but the strategies that they might employ may look a little bit different than younger adults. How about retaining of memory? So I have nothing shocking here. Basically, sleep, exercise, and relaxation, they're all pretty darn good for memory retention. And especially sleep. There's beautiful work now showing that when we sleep, our brains continue to be active. And especially after learning, some of the neural features almost rehearse what we learned during sleep. So I can't emphasize getting exercise and sleep and actually taking a break, allowing our brains just to be, all are very good things we can do. Now, some of the active strategies that we all should be employing when it comes to memorization is making the information meaningful, so evoking sensory information, evoking one's personal experiences always helps with memory. Organizing the information, visualizing it, storytelling. So unfortunately, very passive rehearsal doesn't do that much for actually encoding that memory. We actually do need to put in a little bit of work. And then there's certain strategies you can certainly employ, such as keeping a routine, organizing the information, things that you kind of already know, but might want to think about when it comes to sort of improving how you perform on a day-to-day basis. Now, there are some experimental interventions that I'll end with, just to highlight that scientists are doing something when it comes to memory. So one of the things that we do in our laboratory as a potential sort of intervention for enhancing cognition is something called transcranial direct current stimulation. It sounds a little bit maybe scary. It's not. I've done it many times in my lab, but you do end up putting 2 electrodes generally on your head, and you get a little bit of a current. It's a low-level current, but the idea is that we can maybe modify the underlying brain regions and perhaps enhance especially activation in those frontal regions. So to date, the science is not compelling. There's some work showing that it has some very transient benefits, so there are clinical trials now to see if we keep on administering this over and over again, can it have some more lasting impacts? And then there's brain training, so doing computerized exercises to perhaps enhance cognition. I mean, there's some evidence that perhaps it can help, but generally it tends to help the task that you are learning. So you become very good at this one game you're playing, but does that really translate to anything kind of daily? I think we kind of have some work cut out. And then one of the things that we're looking at in the lab is a sort of a herbal supplement, I guess you can say, which is CDP choline, which has shown some promise of enhancing cognition. So what we're currently doing is giving a person the supplement and seeing whether that has any effects on the brain. I'm always a little bit skeptical with supplements, but there was data to suggest that maybe there's something there, so we owe it to the public to explore this. And on that, I will end. I'm not sure if I have time for a question or two, or... Okay, we'll do it at the end. So thank you so much.
Thank you. That was very informative, Natalia. So if your stream of thoughts generated a question, please hold on to it, keep it in your synapses, transfer it into the hippocampus where short-term memory and long-term memory function reside. Hold on to it, visualize it, and at the end, all the speakers will be up here and you can hopefully retrieve it and pose your question. So you just heard a little bit of an overview as to one really important brain function, namely that of memory. But that's just one of many, many different cognitive functions that have evolved with our evolution as humans. There's many, many more. And so when family members or patients develop problems with their cognition, and they're either going to be seen based on the first presenting symptom by the family doctor or by a neurologist or psychiatrist, very often we want to quantify and better characterize what is wrong with the thinking part of the brain, and we have to employ very sensitive tools to characterize what's wrong. And we wouldn't be able to do this without the help of neuropsychologists who have studied how to properly structure and assess somebody's cognition and define the thinking problems, which then help us to identify the diagnosis and treatment. And so it's with that in mind that the organizers have invited Dr. Mark Coates to speak to you next. Mark is a practicing neuropsychologist who is here in the city, and he told me earlier that he's one of 30 colleagues that work together to try to assess adults for cognitive dysfunction. And he will tell you a little bit about the wonderful array of neuropsychological tests that's being employed. So, addressing memory and as memory relates to dementia, please let us welcome Dr. Mark Coates.
Thank you very much for that introduction. I'm not used to talking with a microphone. This is going to be a little fun. As you just heard, I work as a practicing clinical neuropsychologist. So day in and day out, what I see is people who are saying that they have memory problems. Really, almost everyone who comes to see me identifies memory as an issue. Once I do the assessment, I often learn that there's other things that are actually the issue, and that's really what I want to talk about today, kind of filling out the picture of what else could be causing apparent memory problems and what someone would identify as being a memory problem, when perhaps it may be something else, and that might lead to different diagnoses or prognosis. Just quickly about myself, the place where I work is the Center for Interpersonal Relationships, where we are mostly clinical psychologists, but also there are a handful of us as neuropsychologists working across the age range. We even have people that work with kids. I'm a part-time professor at University of Ottawa, where I teach the psychopathology class, which is all about assessment and diagnosis. Honestly, I do that for a hobby. I love teaching. And then I'm also very, very lucky to be affiliated with the Geriatric Psychiatry Community Services of Ottawa. They are a wonderful organization. I'm just a volunteer member on the board, but these guys are miracle workers, so I had to give them a shout out. When I do an assessment of someone's cognitive functioning, I really have three questions in mind, and these are the three that I need an answer to before I can say what's going on with this individual person. First off, they're saying they have a deficit, but is this deficit significant enough that we would consider it to be potentially diagnosable or evidence of a memory disorder? So is the deficit or problem that they have significant enough? If it is a memory condition, what aspect of memory is impaired? And you just heard about many types of memory, and I'm actually going to add one more as well. Or is it something else? Is it some other cognitive function that sort of supports memory, that for that particular person, that supporting cognitive function is not working too well? So our first question then is it a significant decline? That's my very first question that I want to have answered, because it is normal for all of us to have memory lapses. As you heard, especially as we get older, once we get to be about 65 years old, it's getting close to half of people, 40% or so, who will report some kind of memory difficulties. But even at any age, people will report occasional lapses. It happens, right? We forget where we left our keys, and we forgot which was the road, what street am I supposed to turn on, or what was that phone number? We all make these kind of mistakes. But to really find out if that is something that is serious enough for us to consider a memory disorder, first we ask questions, and then we do a formal cognitive assessment. These are some of the ways that I would ask questions to try to find which category generally a person would fall under. That normal memory side, the normal memory lapses side, is things like someone told me their phone number and I forgot them. I forgot their phone number, or I recognize a couple people here, but where do I recognize you from? I'm not 100% sure. Someone that I saw years ago, perhaps. Needing to remember to do an appointment that only occurs every 6 months. Well, I'm sorry, I'm going to need to write that down in my phone. Right? I'm not going to remember my dentist appointment unless it's in my smartphone. Those types of lapses in memory are quite normal, and although they can be very distressing for people, we all want to be careful not to consider those to be diagnosable memory disorders or any kind of evidence for dementia. On the other hand, the ones on the right here are things where we might get pretty concerned about. If your family is the one that's worried about your memory, versus you being the one that's worried about your memory, that's a really strong giveaway much of the time. When a person themselves is concerned about their memory, they usually have quite a degree of insight into the difficulties they're having. But when folks don't recognize that they're having these memory difficulties, but their family does, then that's often a cause of concern. But similarly, now we're forgetting something that happened just yesterday, or forgetting the name of a close friend or a family member, having trouble navigating in your own neighborhood versus navigating in a town that you're not very familiar with. So there's a pretty clear distinction. This makes it sound kind of cut and dry between what's pathological and what's normal. But of course, there's a huge gray zone between these two, and that's kind of where we would come in with our more formal assessment measures. Also in that gray zone is this term you've heard called mild cognitive impairment, right? Which refers to someone who has objective memory difficulties and other cognitive difficulties on our objective tests, but that they're still able to function relatively well at home. They may be relying more on calendars, or they may be relying a lot more on the spouse than they used to, but they're not doing dangerous things like leaving the stove on, and they're not doing, making huge mistakes in purchasing or other cognitive mistakes like that. The reason why that's an important thing to consider is that a lot of folks who have mild cognitive impairment will go on to develop dementia later on. So it's an important thing to assess for and consider, so that we can give someone a bit of a roadmap of where things may be going, or give them some sense of what their risk is going forward. Neuropsychologists, we kind of live in that gray zone that I've been talking about, between normal aging and mild cognitive impairment and formal memory disorders. Much of the time when it's a moderate or severe dementia, honestly, I'm not needed. You don't need to go for a neuropsychological assessment when someone is that far advanced in terms of dementia, because the symptoms are pretty obvious. They don't really need to be formally assessed by that point. The physician doing the screening would do that in their office fairly quickly, so there's no need to invest in the time and effort and resources for a neuropsychological assessment. We're usually involved when the diagnosis is unclear, when we're not sure if it's this dementia or that dementia, or when it's relatively early on in the course of a condition. That's where we can really help out and give some better ideas to what's going on and what perhaps we can do to help this person. Neuropsychological assessments can take a long time. Some of the quicker ones, like they do at the Bruyère Memory program, they'll do it in about 4 hours or so. I also do some of those that are very brief like that, but a full comprehensive neuropsychological assessment may take closer to 8 hours, so it's a pretty demanding chunk of time. And we would assess all of these aspects of cognitive functioning that I'm going to talk about in great detail for all of them. Not only that, not only do we assess memory and other aspects of cognition, we're also curious about emotional functioning, people's behavior, what the spouse or family member or friends have noticed. So we try to pull in information from all kinds of different places and reach a conclusion about the person's diagnosis on the basis of all that information. Now, I'm just going to quickly show you some examples of some of what the tests would look like. Now, I should say none of these are actual neuropsychological tests, but they're ones that I feel give a pretty reasonable idea of what our tests would look like and what you might encounter if you or someone you know were to be involved in a neuropsychological assessment. Now, you can do all of these in your head. I'm not going to ask for hands up for answers, but do feel free. If you want to shout out an answer, then that's fine too. So we just heard about the different types of memory that we might be interested in, like semantic memory and episodic memory. As neuropsychologists, one of our important distinctions is whether it is verbal memory difficulties that a person may be having or visual memory difficulties. And just that distinction tells us a lot about where in the brain the pathology may be happening. We also want to know where in the stage of the memory process are they having a dysfunction, if any. Are they having trouble learning? Or maybe they can get it in there just fine, but they can't hold on to it. Or maybe they can get the memory in, they can form a new memory and hold on to it, but they can't pull it out when they need to. And all of those we can assess just by how we ask our questions in these formal memory tests. So, for example, that's a little small. I may show a figure. Okay, a figure that's sort of an abstract image of something, and then say, I want you to remember that because I'm going to ask you to draw it later on, or I'm going to ask you to remember what that thing looks like. That would be a very simple visual memory task. For a verbal memory task, I might give a short story. Angela, I would usually read this. I would not have someone read it themselves, but Angela's car would not start, and she had to call a tow truck. She emailed work to say that she would be 1 hour late. Her boss understood. And please remember that, because a while later, I'm going to ask you to repeat that for me. Okay? One other type of memory that I love to talk about because we don't talk about it enough is something called prospective memory. Prospective memory is memory for future intentions. And you all do this over and over again, every day, and in fact, all kinds of times a day. This is remembering to do something in the future. Okay, and it's not quite the same as these other aspects of what we really think of as memory. Prospective memory is, I meant to do something tomorrow, and then I, in fact, remember to do that thing tomorrow. It could be something long-term, like I need to go to the dentist next month, or it could be something really, really short-term, like once I hang up the phone, I have to go back to the stove and turn off the oven, or turn off the stove. Okay, either of those things are prospective memory tasks. Now, we see prospective memory impairments in so many neuropsychological conditions, not just dementia, although that's certainly the case very often there, but also attention deficit hyperactivity disorder, depression, other psychiatric disorders, traumatic brain injury, very, very prominent difficulty there. So as an example of a prospective memory task, I would ask you to do something later on. So when I reach the slide that says making the diagnosis, I just want you to raise your hand. Okay, that's a prospective memory task. I'll be honest, we don't assess this enough. This is why I'm trying to sort of stimulate talking about prospective memory. It's so vital to our day-to-day, and it's one of the most distressing things when we make errors about this. Okay. One of the areas that we assess the heck out of when we do a neuropsychological assessment is one's ability to pay attention and concentrate on a task. If you can't pay attention and concentrate, you can't do that initial memory formation, and you can't do language-based tasks, and you can't do more complicated problem-solving tasks. All of these things at their very basic rely on being able to pay attention and being able to concentrate. So this is something that we assess in great, great depth. Just as an example of one of these tests, probably the most famous test in all of neuropsychology is called the Stroop test, or the Stroop interference test. And in this one, I would ask you to say the color that the ink is printed in and not read the word. Now, if you were just reading the word, you could just say purple, orange, blue, red, purple, black, green, yellow. That's not even hard. But trying to say the ink color would be considerably harder. And even if you're doing it in your head right now, you can imagine how much more concentration that would take to complete that quickly. Okay? And people who have attention and concentration difficulties would have a real hard time doing a measure like this. We want to assess speech. Very, very often people have what are in fact sort of language processing disorders that they perceive as memory problems because they'll say, I can't remember the word. It could be that the memory for the word is in fact still in their semantic storage. They just have trouble retrieving it, that they have the inability to pull that out when they need it. So we'll do simple tasks like I'm going to read increasingly long sentences and ask you to repeat after me. Or we'll give simple line drawings of pictures, usually starting easy, a picture of a flower, and then we would get to things that are a little bit lower frequency, and then asking things that are relatively hard to identify. And people who have language-based difficulties, including some dementias, would have great difficulty even with some of the fairly simple or more recognizable items of these sort of tests. Next, we're always interested in visual spatial reasoning and functioning, how well you can understand what you see, make sense of what you see, do mental rotation, figure out how things fit together. All of that, although in our day-to-day we don't recognize how much we use it, in fact, it can be quite a problem when there are deficits in this area. This is a really tough one, right? Which of those on the bottom is the same as the one on the top? It's in fact the one on the right, I think. Now, I should say here that these types of measures, we don't do them because we think you're going to be doing puzzles like this in your everyday life, right? We don't think that they exactly map onto the precise functional things that you were doing in your day-to-day. Rather, they're diagnostic tools that tell us about what areas of the brain are likely to be affected, and that tells us then what the pathology is likely to be, and that gives us information about diagnosis and prognosis. But very often when I get to these types of tests is when my clients say, what's this for? I came here for memory. Why are we playing with blocks and doing all these sorts of tasks? But really can be very informative. How fast one processes information, something that does tend to decline a little bit with age, and then especially with certain neuropathologies like stroke. I might ask you on a test like this, circle all the 3s as fast as you can, or cross out all of the even numbers. Some sort of simple task like that. It's not cognitively demanding per se, but it's tough to do it really fast. And we're wanting to compare how fast someone is on a task like this compared to their age peers, so that we can get some sense of whether they are having a harder time with the speed of information processing compared to most people their age. A couple more areas of functioning that I look at. We have this large area called executive functioning that Natalia touched on there about that central executive. Executive functioning is all of that stuff that, that executive assistant would be really, really good at. Planning things out and putting things in order and making sure you're solving problems in the correct way. Mental flexibility, shifting between two tasks, these are all really, really difficult things to do. But often this is the impairment that underlies an actual memory disorder or difficulties with remembering things. So some simple executive functioning tasks that I might give would be something like, tell me how you make a peanut butter and jam sandwich. Okay, it seems very easy for most, but it actually requires you to think about all the various steps you would do and be able to explain them in the correct sequence, and would give us some idea if you're able to put things in the right order. A simple game like 20 questions, that I'm thinking of something, you have to ask me yes or no questions to guess what it is. Well, that requires you to listen to my feedback, take that into account, update your idea of what it could be. Requires a lot of mental flexibility, strategic planning. So it's actually a remarkably good measure of executive functioning. Another famous one is drawing a clock. We have many, many, many executive functioning measures. So now we've had a bit of time, and I might ask you to draw that figure that I showed you before. And I'm not going to ask you to do that, but if you could imagine, some of you are doing it, I see, that's great. Okay, or maybe I could do this, and I could show you the two of them and say which one was the one that you saw before. Left, okay, very good, yeah. And you can see how that might give us some idea as to whether your retrieval is a little bit stronger or weaker, okay? Tell me that story that I read to you before. Okay, that might be a little bit more difficult. It was a bit more involved. Do you want to answer?
Battery in the car.
Battery in the car, very good, yeah. Okay.
Called her boss and he said not a problem.
Called her boss, said not a problem? Good, so a couple of the big aspects of it. So why was she late? Who did she call? I might ask with cues, like what was her name, this sort of thing. Oh, you emailed her boss. Okay, look, two people at the back there put up their hands because we've now got to the making the diagnosis slide, okay, and that was your prospective memory test. Now, isn't that fascinating how quickly, now maybe some of you fought to do it but didn't feel comfortable raising your hand, that's fine. But isn't it fascinating how hard it is to remember to do something that you had planned to do in the future? So when it comes down to my job, I love my job because I get to be kind of a scientist and a detective at the same time. We meet extensively with the family, if we can, and the client themselves. We ask them all these questions, and that lets us form a hypothesis about what the brain disorder or memory disorder could be. And then we take out our formal tests, and we actually assess that to test that hypothesis. So it's a great little scientific process with a single person involved every single time that we work with someone. One of the most key things that we have to do as people making diagnosis that's very often forgotten is that not only do we have to figure out what it is, but we have to make a strong case for what it is not. So we have to be able to say, in order to conclude that it is in fact dementia, we would have to say it's not head injury, it's not because of substance abuse, it's not because of depression, it's not because of seizures, it's not because of a systemic medical condition like liver disorder or kidney disorder, it's not because they weren't trying on the test because they didn't like me. You know, we would have to make sure that we can rule out a whole bunch of different things before we would be comfortable to go ahead and make that diagnosis. I've sort of already said that first part there, but I think the point that I'll leave you with is that the more we understand someone's unique pattern of strengths and weaknesses, the specific areas of cognitive functioning that are impaired and the ones that are working well, that lets us make the best diagnosis, which lets us make the best prognosis for them, and lets us most effectively decide what treatment might be best for that person or what kind of assistance they might need. And that's why we do what we do. So thank you very much.
Thank you, Mark. That was wonderful. As I said earlier, please hold your thought and put it into your memory storage center. you heard from Mark that this takes 4 to 8 hours to go through thorough, complex, and comprehensive neuropsychological testing. This is why I personally always worry when I spend 20 minutes with a neuropsychologist whether they can already tell how impaired I am. And so, but Mark said something really important that defines a lot of what we do in the area of brain research and mental health and brain health. Namely, he referred to himself as being a detective and a scientist at the same time. And I think this is absolutely true. When we work with patients, we try to be the detective that contributes to finding out what's wrong with the person in front of us. And then as a scientist, we try to put it in the proper context and weigh the pros and cons, how likely or unlikely it is. And I think this is a good segue also to our next speaker, Dr. Andrew Frank, who is a clinical investigator at Elizabeth Bruyère. He's the director of the memory program. He has trained as a neurologist and then went to the Mayo Clinic and did extra training in behavioral neurology and cognitive neurology. And the reason why I think this was a good segue is because of two things. One is he's playing the detective, as just Mark summarized, every day. And on the second level, I also connect with him as a scientist. So on the first level, I also find him very generous when I refer patients to him because I get stuck and I don't know what the precise diagnosis is. He's been very accommodating to see a number of patients that I sent to him. But the second thing is, when I trained in Alzheimer's disease many years ago on some of the molecular pathology aspects of Alzheimer's disease, I was dreaming of a therapy that's related to the work that we've done in the future. And it's a scientist like Andrew who's actually testing this in the clinical setting with patients, what we many years ago worked on in cells and in animals. He's actually testing scientifically how we can treat people with Alzheimer's dementia, for instance, and how can we possibly interfere with the progression of the illness. So it's with great pleasure to introduce Dr. Andrew Frank this evening. Thank you.
And I don't deserve any applause yet, in fact, because we're not there yet. And I know that many people would feel that that is impossible, a cure for Alzheimer's. But if we don't shoot for the impossible, where will we get? We won't get anywhere. And I believe that if we keep shooting for that goal, we'll get closer. And I want to talk to you about that tonight. And that's about what my discussion for the future of Alzheimer's disease is. Where are we getting in that quest? And I've been so privileged to work with Mr. and Mrs. Suter, whom you saw in the opening video. And you could see that it's frustrating. Some of those, getting stuck on that word is so frustrating. And yet you're doing it, you're coping together. And I just wanted to acknowledge what a privilege it is for me to work with both of you together. Your husband's helping you cope with that. And when we are doing our clinic, in fact, you're in one of our clinical trials, as Dr. Schlossmacher just mentioned. We're testing medicines that try to remove the cause of Alzheimer's. And we'll talk about that. And you're in one of those trials. We want to thank you for that, both of you. If I can just acknowledge you and thank you again for that. It's a true gift. It's wonderful working with you. And I want to talk about the trial that you're in as well as how we got there, and why we're testing medicines that we hope will one day treat Alzheimer's disease. As Madame de Rosier mentioned, our population is aging. And as we get older, the risk for Alzheimer's goes up. In fact, with each 5-year interval after the age of 65, the risk doubles. So that between age 65 and 70, the risk is 2.5%. Between 70 and 75%, it's 5%, and so on. It continues to double. That between age 85 and 90, it's a 40% risk of having Alzheimer's and dementia. So over 90, it's over half. And so our goal is to try to remove this disabling condition from our society, so that people can live a full life without having to experience this 5 to 10 year protracted decline, which Nancy Reagan once called the long goodbye. We want to remove that from our society so we can live a full life without this kind of disability. In Canada, it's 500,000 individuals affected today, but in the next 15 years, this will nearly double. mostly because of the aging population. And the economic, of course, it affects individuals and family members, as we said, but it affects all of society at a cost of enormous cost monetarily. So this is a curve, a projection of the future that we want to change very badly. The disease itself is named after Dr. Alzheimer. Over 100 years ago, he was a German psychiatrist and pathologist who saw a woman admitted to the asylum for insanity. We don't use these words anymore. But he was convinced that it must be a physical change in the brain that was affecting her mind, her memory. And so when she died, he looked at her brain under the microscope and saw what we're going to talk about tonight. These proteins were building up in the brain. He was the first one to see them. Amyloid plaques and tau tangles. And since he was the first one to describe them, the disease bears his name to this day. And Dr. Schlossmacher, who is our host tonight, was instrumental in understanding these proteins. And it's these very proteins which we believe are causing the brain damage that leads to the memory loss that we heard about tonight in the hippocampus. Is there a way to remove these proteins, the proteins that Alzheimer first described, now over 100 years later? Can we remove those proteins, perhaps at the earliest stage, so that we can prevent people from ever having to experience this dementia progression? And that is what we're testing right now at Bruyère and around the world, medicines that can remove these proteins. And that is the hope. Amyloid itself, and again, Dr. Schlossmacher's work is central to this, is made, amyloid, this protein in the brain that builds up as plaques, is formed at the wrapping around each of our brain cells. And we have tens of billions of brain cells connected to each other by tiny star projections, synapses to others, to their neighbors, which can then connect to the other side of the brain by longer projections. It's almost like each of our billions of brain cells, all microscopic, are a tiny computer connected to all the other neighbors, almost like an internet across the entire world. But this world is just our brain inside our skull. But when 10 billion computers are talking to each other, together they create nothing less than human consciousness. And all of our thoughts and memories and words and art and philosophy, this is what our brain can do with all of these brain cells connected to each other. But around each of these little tiny computers, these proteins are made. They may start off as helpful, but when there's too much of it, they become toxic. And we're not exactly sure why they start building up. It could be that as we get older, the clearance, almost like the garbage removal system, may start to slow down and these proteins start to build up to toxic levels. Just imagine in Ottawa, and I guess if Mayor Watson was here, I'd say, just imagine if the garbage system stopped working in Ottawa, how quickly the city would break down if we lost our garbage removal system. And what if that's happening in the brain? And what if we can help that system and save all of those precious brain cells and their connections? Can we change the future? And so, as these little tiny proteins are made around each of the little computers, they start to build up and they start to stick to each other. And these tiny collections are toxic to the brain. And if they damage the brain cells, they can't connect to their neighbors anymore. And if you can't connect the brain cells, how can you make that memory? It's a physical, it's not that someone's not trying, it's that there's no physical connection made when information enters the brain. So how could it be remembered? Because we need that physical connection in the brain for the memory to be formed. Eventually, the collections form into bigger collections, which are the plaques that Alzheimer described 100 years ago. And going deeper, factors like age, like we said, as well as genetic factors, and you brought that up. And if I may touch on that now, genetics in Alzheimer's do play a role. When it's in our family, our risk does go up. But here's the key. The risk goes up only by a small amount. It's not inevitable for all family members to be affected. It's not the case. And so, yes, our risk goes up when it's in our family, and it's in my family too. But it's not an astronomical jump in risk, just a small. So it's not worth worrying about every day. But what it is worth doing is having a healthy life and keeping good circulation in our bodies to try to clear out those proteins through physical activity, mental activity, socializing, working in provincial politics. Wait a minute, that might be the other. Okay. But staying active and not worrying, because the genetics are not the whole story. But age is part of it. And our environment. What about the air we breathe? Our water, our food supply, and our lifestyle. They all play a role in how much amyloid protein we make that goes into the plaques. And then it's believed that when the plaques build up, they trigger that second protein, tau it's called, T-A-U, that forms tangles, plaques and tangles, which are clearly toxic to the brain. First seen by Dr. Alzheimer's 100 years ago, and with us today. As the brain cells are damaged and destroyed by the amyloid and tau, they are lost, and the result is dementia, which means memory loss and loss of independence. That's what dementia means. Dementia is memory loss and loss of independence. Alzheimer's, is that the same as dementia? Not quite. Alzheimer's disease refers to the proteins, and dementia is the result. And so if we want to get to the cause of this, we want to go at Alzheimer's, and we want to go at those proteins. Today, we have new scanning technology so that we can see those proteins in the brain while someone is very much alive. Previously, one could only see if one had those proteins after someone had passed away and there was an autopsy. But now, with new technologies, a PET scan, you can see the protein in someone who's very much alive and see if that amyloid is there and make the diagnosis more clearly. And you can bet if we can see that protein, we're going to want to get rid of that protein. And so we use these scans in our research when we begin a clinical trial. And here is another PET scan. The first picture was of, and actually if I may go back, I apologize if I wasn't clear. On the left is a dark scan. That means there's no amyloid there. That's a normal control. But look on the right, all lighting up in red. That's the protein on a scan. That is the problem. And that is someone with Alzheimer's disease. And here, the next picture, it's like a spinning image, lighting up on the sides. This is a tau PET scan. It's a PET scan showing the tau protein, the other one. This is very new technology. And look, and it's hard to see the exact shape of the head there, but you can sort of see that that color, the red, is on the sides of the brain. The temples, what are called the temporal lobes, the temples. And that's where the tau is building up. And sure enough, in those temporal lobes, that's where the hippocampus is. That's where the new memories are made. And that's why it's so hard to make memories in Alzheimer's, because the tau is building up in the temporal lobes, destroying it. Can we get that tau out of there? That's what we're doing. But what do we have today? Before we finish, we're going to talk about the research, but do we have any medicines today for Alzheimer's? Yes, we do. And it's right here. These medicines for mild and moderate symptoms help, but only just a little. They only help modestly. They're worth trying, but they're not enough. And everyone knows we don't have enough. Like, for example, this is what it looks like over 5 years with the treatment I'm talking about, with the treatment we have today. The red dots show memory in someone who has medicine, who's getting medicine. And the white line are individuals who receive no medicine. You can see the red dots go up a little bit, just at the very beginning. I don't know if I can use a pointer, but just at the very left, the red dots go up a little bit, and then they start to decline. So the medicines give a bit of a boost right at the beginning, but look how the condition still gets worse, and the memory still goes downhill, despite the medication. But it's a little better than the white line, which is no medicine. So that's why the medicine is worth trying. But look how it doesn't arrest the condition. Someone still gets worse over 5 years. So the medicine is worth trying, but we need more. We desperately need more. And the medicines help a little bit and may cause side effects such as stomach upset. So I'm watching for these side effects every day in my practice. Can we prevent Alzheimer's in the first place? Only to a degree. We can't prevent all cases. But if we have a healthy life, managing what are called vascular risk factors, these are blood flow risk factors. You've heard about these: blood pressure, cholesterol, diabetes, smoking cessation, loss of excess weight. These are all the things that we use to try to prevent heart attack and stroke. They also help prevent Alzheimer's and dementia through circulation, clearance of protein through circulation. But also, as I mentioned, mental and physical activity, social involvement, and a balanced diet, perhaps even the Mediterranean diet. That's the one with red wine. But also, fruits and vegetables, whole grains, and the light meats, poultry and oily fish. All recommended to try to decrease risk of Alzheimer's. But we can't prevent every case. Hence the hope for the future. Can we attack those proteins, the amyloid and tau? There's a huge effort worldwide to this day trying to find these new treatments, attempting to remove the amyloid and tau protein, which is Alzheimer's disease and leads to dementia. The plaques and the tangles. For example, antibodies. What are antibodies? Antibodies are also tiny proteins. Our body makes them to stick to and neutralize infections, such as bacteria and virus. But how about an antibody that we can make, or rather, that can be created, that doesn't attack a virus, but instead attacks another protein? And sure enough, an antibody has been developed which attacks the amyloid protein and targets it for removal. And so to this very day, we are in fact, the suitors are part of a clinical trial testing an antibody that sticks to that protein and targets it for removal. And the only question that needs to be answered is, will that help memory? Will that prevent Alzheimer's and dementia? And can we start it at the earliest possible phase, before the damage is done? Phase 3 studies are currently underway. What are phase 3 studies? Are the final phase of human studies, meaning they're large and across the world. And if they're successful, that medicine will be on the market in pharmacies and hospitals. So these studies have reached the final phase. And we're testing them today, right here in Ottawa. The anti-tau antibody. This has been much more difficult to develop. A successful antibody that targets tau, the other major protein, the tangles. This has just entered phase 2, the penultimate, the second to last phase, and has begun recruiting here in Ottawa just last month, in September. We began recruiting for the phase 2 study of an anti-tau antibody, and I'm so pleased that this study has finally come to us so that we can target not just the amyloid, but the tau as well. And my great hope is that one of these treatments, or more than one, will show benefit and will be available to all of society. We're investigating oral medicines. In fact, the antibodies are more intravenous. Oral medicines, the secretase inhibitors, which prevent amyloid from being formed in the first place. The antibodies clear what is there. These medicines prevent it from being formed in the first place. Nerve growth factors are being tested around the world, and stem cell research that has long been blocked by ethical concerns about having to use embryonic embryos. No longer, stem cells can now be created from our own body. And if those stem cells, stem cells are cells that become different kinds of cells. So if injected into the brain, they would become brand new neurons, meaning brain cells, new computers in the brain. If, for example, we could use an anti-amyloid treatment to put out the fire, we could use stem cells to plant new trees and bring back the forest of the brain, those connections, to keep the memory and all of those precious abilities we've talked about tonight. Also under investigation right here in Ottawa, we're testing computerized cognitive tests. Simple interface, for sure, that might be more sensitive than those paper and pencil testing. If we use the power of a computer, we might be able to detect the memory loss even earlier, because early detection is so important to us, because that's when we want to start the treatments that remove the proteins. We're also testing at-home cognitive training, mental activities with a tablet game. This has all been developed right here in Ottawa to try to keep the brain active at home and also measure performance at home instead of just in the office. And lastly, right here in Ottawa, developing technologies for what's called the smart home. That's like a home wired to the internet almost, with sensors throughout the house, not to be intrusive, but rather to monitor safety, so that individuals can live at home, in their own home, safely, with memory loss, as long as possible. Again, using technology. All of these research avenues that we've talked about tonight, underway around the world and right here in Ottawa. Bruyère has partnered, as you've heard, with U Ottawa in a wonderful collaboration to move all of these research platforms forward as quickly as possible. And so to conclude, we talked about dementia and Alzheimer's disease, the memory loss, the loss of independence, and Alzheimer's, the buildup of amyloid and tau. It's a threat to us, ourselves and our family members and all of society, especially in the next generation, with the aging population. And we have current treatments like the ones I showed. They help a little bit, but only modestly. They may delay time to serious disability by a bit, but they're not enough. We need more. And to help our risk, we manage vascular factors like blood pressure, cholesterol, diabetes, and a healthy lifestyle is highly recommended. And we are now testing treatment that removes the underlying amyloid and tau protein, which we will hope will get at the root cause and make a real difference. And we truly pray that new treatment options may be available, maybe even within the next 3 years. But no matter what happens, we'll keep testing new treatments so that the future of this condition is a lot brighter. Thank you very much.
Thank you, Andrew, for that wonderful journey through Alzheimer's disease and its development and its clinical approaches. So I hope you all remembered and stored your questions. So I'm going to ask all the speakers now to come to the podium. And we have about 10-15 minutes for questions and answers. And we have helpers that will hand out microphones. So please wait. I'm going to identify who's going to ask the next questions. You'll get a microphone and then just go for it. Okay? Why don't you come up, please? Okay, what's the first burning question? Over there, this young lady. Very young.
Hi there. My question relates to the connection between brain injury and Alzheimer's. We do sometimes hear that there is a connection, particularly around concussion, and I wonder if one of you could comment on that.
Should we start maybe with Mark and then with Andrew?
Is this on? Is this on? Okay, there we go. Yeah, there is research that's coming out now that says that a history of concussions can make one more vulnerable to Alzheimer's-like condition. And whether it is exactly the same as what we heard about in terms of Alzheimer's, we're not quite sure yet. It's still a pretty new area of research that we're having more and more access to, unfortunately, as some of these conditions develop in retired hockey players and football players. Usually what we're talking about, as far as I know in the research, is a history of multiple head injuries. Having a single head injury could perhaps make one's brain a little more vulnerable, but it is not going to be likely related to that, what do they call it, chronic traumatic encephalopathy. It's really where this is happening over and over and over again. As I say, hockey players, boxers, football players, lacrosse players, soccer players, perhaps some risky occupations, but for the most part, that's where we would be concerned.
Andrew, do you want to add anything to that?
Thank you for that question, and you're right, Mark. This CTE, chronic traumatic encephalopathy, it's probably not exactly the same thing, but I can tell you this, is that if we sustain a head injury, guess what happens over the next 12 to 24 hours? The amyloid levels of the brain spike. It's almost like the amyloid might be working to help a little bit. In that initial phase, at the low levels, the amyloid might even be there to help the brain. But if you have enough of those injuries, there might be a buildup of that amyloid, and that might start the cascade towards dementia.
May I just interject here for a moment, because we have Madame Desrosiers here. I have to say, as a neurologist and as a brain researcher, I'm really appalled how in the province of Ontario we stage octagon fights that are where the sole purpose is to hit the opponent's head, cause brain injury, to knock him or her to the ground. And so I think that this is something that as a civilized society I have a very hard time accepting that. And I think we as a community, actually, we have to stand up more and say this is barbaric and we have to do something about it. So I apologize, I don't mean to hijack that question, but it raises such an important issue. And when you mention chronic traumatic encephalopathy, we have to do better together with our politicians to not have millions come to any big city to watch that spectacle, but to actually end it, even though it's a loss of money. Anyhow, next question. Yes, ma'am.
Could you explain the differences between Alzheimer's and vascular dementia?
Do you want to do that, Andrew?
Thank you. Wonderful. Alzheimer's disease is amyloid and tau. Vascular dementia is due to stroke, and which is a blockage of blood flow, decreased oxygen. And there's no question that can cause dementia, that can cause memory loss, that can cause loss of independence. We call that vascular dementia. And because as we age, there is a natural decreased circulation, almost everyone who develops Alzheimer's also has some vascular change as well. And so we call that a mixed dementia, meaning both the protein buildup and the decreased blood flow of vascular and stroke. They're often mixing together, conspiring almost to take away memory.
There's another question in the back. The lady in the purple sweater.
I had a question, but I also have a follow-up to what was just said.
Can you just speak up a little bit so that we can hear you better?
Yeah, I have a question, but I also have a follow-up to what was just said. Is that okay? You mentioned brain injury. There are several of us in the room tonight who have brain injuries. You mentioned the spike in amyloid and that triggering something. Is that something that we should worry about over a certain period of time? Or it happens right away? Or
it's right away, and then the amyloid's cleared away.
Okay.
So that's what amyloid should be, cleared away.
Got it.
It's only the multiple impacts that can cause this buildup.
Okay, and on the clearing away, several of us have heard about sleep and this link to, I don't know, some clearing during sleep. Is that something that is an issue?
It's real, if I may jump in on that. It's real. Sleep is a critical human function. And during sleep, proteins are cleared. During exercise, proteins are cleared. That's why we exercise. And we sleep to clear proteins. And so that's why I'm always testing. I'm ordering a lot of sleep studies because I'm checking for sleep apnea or other sleep disorders that might interfere with sleep because good sleep is so important to health and to memory.
Yes, ma'am.
Hi, my question has to do with prevention. And do you have anything to say about meditation, mindfulness on like helping to prevent and or impact the removal of the plaque? You know, especially mentioning neurons that fire together, wire together. There's evidence-based research that talks about long-term meditators that have really good memory. So is there anything that you can add to that?
I think my mic works, yes. So yes, there is some evidence of that. Some of it really comes from a very select group of individuals that are sort of known to be kind of guru meditators who also minimize a lot of stressors and adhere to a specific type of diet and generally come from a community, generally religious, but so there's other factors that it's always very difficult to tease apart. Is it the meditation or is it something else? But there is now growing research on the benefits of yoga, for instance, in terms of promoting good mental health. And again, my work is more with mental health disorders, and one of the things we do is specifically look at not meditation, but actually exercise, because the evidence for that is a little bit stronger, and specifically cardiac exercise. So we do know that very, not super intensive, but one that gets your heart going quite regularly. There's been numerous trials now that show that actually has very beneficial effects on the brain. So we're generally talking about not individuals with a dementia, but individuals with various kind of psychiatric problems. But we can show that there's actually promotion of certain neuronal factors that kind of bathe our neurons in these really good sort of soups that promote neuronal connections, neuronal growth, get more oxygen going. And we know that is so critical. So meditation, the theory is that it may also have one of those sort of pro-cognitive effects as well. But the evidence is a little bit more difficult to uncouple from all these other factors.
May I jump in there on that one as well? Speaking of coming together to care for the aging brain, one of the best uses of mindfulness and meditation type practices is with caregivers. If we can teach caregivers to be mindful of how they are reacting to their spouse or their mom or dad or someone else, that can have tremendous impact. And one more shout out for GPCSO, they actually have case managers who go out into the community and work with just caregivers, just teaching them mindfulness meditation, and I've had great success with that. So that's where I would want to put the eggs in that basket. Great question, thank you.
Okay, there's a gentleman in the back.
I suppose my question is to the panel as a whole. This evening I heard reaction to brain stem cell research as positive. On Wednesday evening, when I listened to Dr. Bruyère, he didn't have the same opinion. His comment was, I don't see a lot of future or benefit. I think I'm most probably misquoting him, but the theme was, I'm not going to spend a lot of time there. There are other things that I want to do my research on. To what extent is a single researcher allowed to hijack an agenda? Blasphemous words, but you know where I'm going, as compared to a policy decision or a mission statement by the organization as a whole that says, we want to go in this direction? Or is it because they're two different diseases that my whole comment is irrelevant?
So I think that's an excellent question. To go back to the beginning of your question with respect to Dr. Ari Bruyère, he most likely, the way I understand him, is addressing the issue of, for instance, ALS or Lou Gehrig's disease, where currently there's not a lot of evidence that introducing stem cells into the fluid that essentially surrounds the spinal cord will help the cells to get into the spinal cord and reconnect all the way to the muscles of our arms and legs. I think it's a little bit like thinking you take a new chip and throw it into an old computer and it would do the job as a new computer. We've also learned that this is probably not going to work in Parkinson's disease, but it doesn't mean that it may not be applicable to other diseases. And there's actually different types of stem cells. And in stroke, for instance, there's now trials that have actually shown the benefit of a distinct type. They're called mesenchymal stem cells. They're not nerve cell precursors, they're precursors of other cells, that they actually seem to modify the recovery after stroke. So I think it depends on what disease model before human studies and then what human diseases you try to address. That's part one. And the second part, when you refer to how much can a scientist hijack the idea from another disease, if I understood you correctly. And the way it works in the Western Hemisphere is that we haven't solved these complex diseases. So anybody's idea is for him or her to pursue, as long as he or she is qualified to do research, raises the money to do the research, and does it within the ethical boundaries. But we actually welcome people from other disciplines more and more than we did even 5 or 10 years ago, because we have recognized that people with a completely different skill set, different ideas, and different training records can vastly expand our own understanding of disease and therapeutic approaches. So we would be fools if we were to continue to be dogmatic, to say our consensus is what everybody else has to sign up for, because then we may inhibit research that actually can make meaningful progress. So we have to keep an open mind, and this is why this process of peer review, making your submissions for the next research project be reviewed by your competitors and colleagues, is so important. Definitely in Western democracies. So it's a good question, but one solution doesn't fit every disease, and everybody is welcome to come with new ideas in the scientific arena. Maybe we'll take one last question, and then we have, then I will introduce our speaker who is going to send us off tonight to get some sleep, to make sure that we consolidate all the things we learned today. Okay, one more question.
I have a microphone, if I may ask a question.
Yes, go ahead.
Okay, well, thank you all. And one of the things that I think, Mark, you said that I found very kind of encouraging, and I think it must apply to all of the research and scientists here, and I guess all of us, but is this notion that memory disorders have unique patterns. And I find that assuring, because when if I think of the brain, then I think, oh, if it's not working, it just must be a sludgy mess of chaos. But if the idea that there are unique patterns within disease and that there are still unique patterns of functioning, I find that hopeful. And I just wondered if you have any comments to add to that. How does that work? Like, does that play a major role in how you do research? Is that why interdisciplinary contributions are so helpful? Like, could you comment on that, please?
Yeah, just a quick comment from the clinical side of things, that conveying that diagnosis, I talked about the process of how I get there, but I left out the most important part, which is saying to someone, I believe you have this condition. That is an extremely difficult, extremely meaningful, one of the most important things that I do. In my job, is to make sure that they understand what I'm talking about, what that means for what areas are working and which are not working, what it means for the future. It actually answers a lot of questions, because most of the time people come into it, as you say, thinking there's something sludgy, or there's something that's not quite working, I'm not as sharp as I was, or this generic, there's a problem with my memory. And if we can break it down into the smaller component pieces, I find a lot of people really, really respond well to, okay, now I know which things are working and not working. Now I know what I might be able to do to compensate for those things as they are now. So that's a really, really key part. Thank you so much for asking that.
If I may just quickly jump in, I think you have a really beautiful perspective on it, because sometimes I, as a scientist, view heterogeneous symptoms. So, well, this person is presenting with this person is presenting with this person is presenting with this, yet they're all depressed. That's kind of an obstacle for me as a scientist, because I sort of want to put everybody in a box. But I think what you're pointing out is that sometimes the heterogeneous nature of specific brain-based diseases can actually be quite hopeful, because what sometimes we don't do a great job of is matching the appropriate treatment based on the symptomatology. We kind of do a one-size-fits-all occasionally, especially in my world of psychiatry. Everybody gets, Prozac because they're depressed. Yet we do have more tools in our toolbox. So I think that's a really lovely perspective. Thank you for that.
Well, on that note, thank you very much for your excellent questions. I would like to take the opportunity to thank our three panelists and speakers for doing an excellent job. When I was trained in... When I was trained during my fellowship, I had a wonderful teacher who was the head of the Cognitive Memory Disorder Unit and the Movement Disorder Unit at the Mass General, Dr. John Grodner. And when I asked him, how do you want me to report to you? He said, ABC, Michael. And I said, ABC? And he said, accuracy, brevity, clarity. And I think all three speakers met that standard tonight. So thank you very much. And they also all told me that they will not retire until they've made a dent into this disease. So in closing, I'd like to introduce you to Dr. Heidi Seistrup. She's going to send us all off. Dr. Heidi Seistrup is the Chief Scientific Officer of the Institut de Recherche Bruyère, the Bruyère Research Institute. She's a force to reckon with. She's an excellent researcher herself. She also has this rare gift that we are never trained in and are not born with, namely administrative and organizational talent. And she has brought wonderful change to the Elizabeth Bruyère. And from our perspective at the Brain and Mind, we are really blessed to have entered this partnership. And anyhow, welcome, Heidi, and the podium is yours.
So I see that people are putting their coats on, so I'm not going to keep you much longer. So I'm just really going to say how excited I am. Every time we have the opportunity to bring our scientists together, our trainees, hopefully you were over there and saw some of the anatomy of your brain. You can actually see what your brain looks like. You can talk to another trainee who's going to show you how your brain functions when you're playing certain types of memory games. It's just so thrilling that we were able to join with the University of Ottawa Brain and Mind. Our students and our trainees, our clinician scientists, our scientists, they learn from each other, they're collaborating together, but more importantly, they really push each other. They're challenging each other every day for all of the work that they're trying to accelerate so that they can let these guys retire, solve the problem of not only Alzheimer's disease, but the many dementias and memory loss problems that are out there. We know that there are over 1 in 5 Canadians who are carers for someone with dementia. There's over 500,000 Canadians who have dementia. And we know that over 50% of Canadians, so half the room here, this is a special audience, so probably more than that, but half the room in any meeting is concerned about dementia. That's a lot of Canadians. So we're really excited about this partnership that we've created with the Brain and Mind at the University of Ottawa. I want to thank Dr. Schlossmacher. I know you saw our video, Dr. Knoffel and Dr. Ismail, who were the co-leads for the memory and cognitive group in the Brain and Mind, and all of the scientists and trainees that we have working on this problem. But most importantly, I think I want to thank the patients and their carers, not only their formal carers, but their informal carers, because they're so incredibly important to the solution. We have people in the audience here who have now given our scientists a new challenge. What's the next problem that they want to solve? And so we're really excited about the initiative, and it's only through the support of our community, that we're going to be able to solve this really important problem. So I want to wish you a wonderful evening. Thank you for spending the evening with us, MPP Desrosiers, and declaring this Brain Health Awareness Week on behalf of the city and Mayor Watson. Drive safe and sleep tonight, exercise, and eat well tomorrow.